Good morning, students. Today's topic is cell organelles. Now, what is this cell organelles? Cell organelles are different substance that are or structures that are present within the cell, and this is covered by a cell membrane. Now, when there is rupture of cell membrane by any trauma or by some chemical methods, these subcellular organelles get exposed. There are different marker enzymes that are present in different subcellular organelles. In mitochondria inner membrane there is ATP synthase. In lysosome there is cathepsin. In Golgi complex there is galactosyl transferase. In microsome there is glucose 6 phosphatase. In cytoplasm there is lactate dehydrogenase. Now coming to different cell organelles one by one. First, nucleus. Nucleus is present in all cells except mature RBC. It contains of two membrane. One is inner membrane. It is also called perinuclear membrane. It contains pore. Outer membrane is continuous with membrane of endoplasmic reticulum. Nucleus contains DNA. Very long DNA complexed with protein forms chromatin. When it is further condensed, chromosome is formed. In nucleus, DNA replication and RNA synthesis occurs. In nucleus, there are light shaded area. This is called nucleolus. In nucleolus, there is RNA processing and ribosome synthesis. Vesicular transport across membrane occurs via endocytosis and exocytosis. Importing and exporting are involved, which is helped by RNA gap protein. Next, endoplasmic reticulum. It is a network of interconnecting membrane covering channels or cisterns. In under electron microscope, this appears just like railway track. In endoplasmic reticulum, protein, glycoprotein and lipoprotein are synthesized. It also helps in detoxification of drugs, and microsomal P450 enzyme helps in this. Endoplasmic reticulum can be of two types, rough and smooth. Rough contains ribosome on cytoplasmic side, which helps in protein synthesis. Next one is Golgi apparatus. It is convergent area of endoplasmic reticulum. It contains flattened smooth membrane and vesicles. Its function is protein sorting, packaging and secretion. Now after these steps occurs, finished products may have different fates. It may pass through surrounding media and resulting in a continuous secretion. It may be integral part of plasma membrane, but these are not secreted. It may also form vesicles for longer storage. These vesicles are ruptured under specific stimulus and secreted upon that. One example is trypsinogen. Trypsinogen is present in pancreatic cells, and when it is stimulated, this membrane rupture and trypsinogen is converted to trypsin. In some connective tissue disorders such as Sjogren's syndrome, there may be presence of anti-Golgi antibodies. Next one is lysosomes. Lysosomes are teeny organelles. It contains bag of enzymes. It has some defensive mechanism. It removes damaged or dangerous particles from cells. Endocytic vesicles and phagosomes fuses with lysosome to form secondary lysosome. Foreign particles are destructed within this. Lysosomal enzymes acts in pH 5. Different lysosomal enzymes are polysaccharide hydrolyzing enzymes such as alpha glucosidase, beta galactosidase, hyaluronidase, aryl sulfatase, etc. protein hydrolysing enzymes such as cathepsin collagenase, nucleic acid hydrolysing enzymes such as ribonuclease deoxyribonuclease, lipid hydrolysing enzymes such as phospholipases. Next organelle is peroxisome. Peroxisome have a granular matrix. It contains peroxidase and catalase enzymes. Now, this peroxidase and catalase removes free radicals which are generated when there is peroxidation of polyunsaturated fatty acids. These free radicals are very dangerous for our bodies. Now, this deficiency of peroxisomal enzymes can lead to different diseases such as adrenoleukodystrophy, Zellweger syndrome. In adrenoleukodystrophy, there is deficiency of peroxisomal matrix protein, and it is characterized by progressive degeneration of liver, kidney and brain. It is a rare autosomal recessive condition, and defect is due to insufficient oxidation of very long chain fatty acid. In Zellweger syndrome, proteins are not transported into peroxisomes. This leads to formation of empty peroxisomes or peroxisomal ghost inside the cells. Protein targeting defects may occur due to this. Next one is mitochondria. They are spherical oval or rod-like bodies about 0.5 to 1 millimeter in diameter and 7 millimeter in length. Erythrocytes do not contain mitochondria. Mitochondria is powerhouse of cells. Energy generated from oxidation of food stored in this as ATP. It contains two membranes, inner membrane and outer membrane. Inner membrane folds to form crista. This inner membrane contains enzymes of electron transport chain. Cytochrome P450 system is present in mitochondrial inner membrane, which is involved in steroidogenesis. Superoxide dismutase is also present in cytosol and mitochondria. Mitochondria also contains specific DNA. The integral inner membrane protein are made by this mitochondrial protein synthesizing machinery. However, majority of proteins, especially of outer membrane, are synthesized under control of cellular DNA. The division of mitochondria is under the command of mitochondrial DNA. Antibiotics, those works by inhibiting bacterial protein synthesis, actually inhibit mitochondrial protein synthesis. Mitochondria also plays a role in apoptosis. Now, let's have some of the functions of different these cell organelles. Nucleus, it helps in DNA replication and transcription. Endoplasmic reticulum, it helps in biosynthesis of proteins, glycoproteins, lipoproteins. It also helps in drug metabolism and synthesis of cholesterol. Golgi body, it helps in maturation of synthesized proteins. Lysosome, it helps in degradation of proteins, carbohydrates, lipids and nucleotides. Mitochondria, it helps in electron transport chain, ATP generation. TCA cycle, that is tricarboxylic acid cycle, beta oxidation of fatty acids, ketone body production, urea synthesis, heme synthesis. These are partly. Gluconeogenesis, this also helps in part. Cytosol, its function is protein synthesis, glycolysis, glycogen metabolism, HMP shunt pathway, fatty acid synthesis, cholesterol synthesis, and part of heme synthesis, urea synthesis, and pyrimidine and purine Synthesis.